We've all seen the preclinical hype around exosomes acting as, native cellular messengers. We've got a custom deep dive just for you today, parsing the June 29th to July 6, 2026 exosome reporting window.
Right, because for years they were supposed to be the ultimate biological delivery vehicles, perfect diagnostics, and regenerative miracles all rolled into one.
Yeah, all rolled into one. But the reality of scaling that biology is notoriously brutal.
Okay, let's unpack this. We are looking at a pivotal week where the exosome field really split into clearer lanes.
It did. What's fascinating here is the tone has fundamentally shifted. It's less about discovering novel biology now and much more about the rigorous discipline of manufacturing.
Right, balancing a massive regulatory win with a pretty harsh real-world reality check. Let's start with where that rigorous engineering is actually paying off. INTENT Biologics just landed an FDA breakthrough therapy designation for their PP biologic.
Right, the shelf-stable platelet-derived exosome product. Exactly, aimed at treating diabetic foot ulcers. But I look at this and think, it's kind of like getting a VIP club line pass. You get to skip the general admission. Wait, but are they actually inside the club yet?
Well,
Especially since the FDA still explicitly states there are 0 approved exosome products on the market.
you're absolutely right to be skeptical about the fast track label, but don't let the lack of full approval overshadow what they actually proved here. This is the strongest U.S. regulatory signal yet for a true exosome biologic.
Because they actually define the active components.
Exactly. They aren't just saying exosomes heal. Platelet-derived exosomes naturally carry specific growth factors and RNA that instruct surrounding tissue to ramp up angiogenesis.
Which is new blood vessel formation, right?
Right, and dial down chronic inflammation. They prove potency because they combined a defined product with those measurable clinical endpoints.
Okay, so controlling the environment in manufacturing works. But here's where it gets really interesting. What happens when you leave that controlled factory floor and try to extract natural exosomes from random patient blood samples in the wild?
Oh, it gets messy.
Yeah, INOVIQ's Exosome ovarian cancer test hit a massive wall here. It's like having a Michelin star recipe but spoiled ingredients.
That is a perfect analogy. The pre-analytics are brutal. In their initial highly controlled 500 sample set, the algorithm was stellar. I mean, they showed a 92.3% sensitivity for stage 1 and 2 cancer.
Wait, really? With 98% specificity, right?
Yes, but when they expanded to a 1,200 sample retrospective cohort drawn from various biobanks,
The whole thing fell apart.
completely, the samples were deemed entirely unsuitable.
This raises an important question, though. Why are these extracellular vesicles, or EVs, so fragile in a real-world setting?
Think of an exosome like an envelope with an address written in delicate ink. If you leave it out in the rain, or in this case, subject it to variable freeze-thaw cycles in different biobanks, the delicate protein markers on the exosome's lipid bilayer just physically shear off or degrade.
So you can have a brilliant algorithm, but if the biomarkers are erased before the sample even hits the lab, the test is blind.
Exactly. It's not a lack of biomarker imagination. It's literally just sample logistics and pre-analytics ruining the signal.
Wow. Which brings up an interesting pivot. If extracting them for diagnostics is that messy, and standalone therapies take years to clear, could exosomes simply act as biological delivery trucks?
Right, like pure cargo vehicles.
Yeah. Spot Biosystems just launched with $40 million to test exactly that, using EVs for gene delivery. They're targeting full-length dystrophin mRNA. Now, for those following the space, Duchenne muscular dystrophy is caused by a genetic lack of this exact protein.
And Spot is reporting they've successfully delivered this mRNA payload in animals, right?
Yep, and they even have early signals in 2 pediatric Duchenne patients. So is this non-viral delivery route the actual safest bet for the industry right now? How does it differ from viral vectors?
Mechanistically, it's brilliant. Traditional viral vectors trigger the body's alarm systems, but exosomes, being native cell messengers, can just slip past the immune system undetected to deliver genetic payloads.
But I have to ask, are we jumping the gun here since the human evidence is still so early?
We are definitely jumping the gun if we declare it a sure thing. The data is extremely preliminary. If we connect this to the bigger picture, the true winners across therapies, diagnostics, and delivery won't just have good biology.
They will be the ones who conquer manufacturing.
Exactly, proving potency and defining their active components.
So what does this all mean for you listening? The exosome field is getting very real, but significantly more disciplined. The gap between promotional claims and approvable products remains large.
The The science is maturing from hype into unglamorous engineering.
Which leaves you with a provocative thought. If the true bottleneck in the exosome revolution isn't the biology itself, but rather mundane things like biobank storage variability and scalable manufacturing, how will that change which companies ultimately survive the clinical trial gauntlet?
Right, will the best biology win or just the best logistics?
Something to mull over on your own. Thank you for diving in with us today.